Good morning and welcome to the PNOC Informational webinar for high-grade glioma. Today our panelists will give an overview of high-grade gliomas, the current treatment landscape and clinical trial development. My name is Bruce Campbell, and in 2013 my wife Alan and I helped co-found PNOC Foundation 3 years after our own son George was diagnosed with an anaplastic ependymoma. At that time, resources like today's webinar and access to this global panel of experts in a group setting didn't exist. We're grateful to have and to share this access with all of you. At the PNOC Foundation, our mission is to urgently raise funds, resources and awareness to support PNOC's research and clinical trials, to ultimately improve outcomes for children or for all children with brain tumors. For those of you joining us for the first time, PNOC is a leading global pediatric cancer consortium with over 40 study sites in the US, Europe, Australia, Israel, India, and Egypt, striving to improve outcomes through a unique collaborative approach, translating the latest findings in brain tumor biology into innovative novel clinical trials and more effective treatments, all whilst maintaining a real sense of speed and urgency. Joining us today is a large, diverse audience of over 100, spanning from those whose children are newly diagnosed to those who may have been dealing with this disease for some time. We want to answer your questions and encourage you all to post those questions in the Q&A versus the chat for our panelists. We'll kick off with an overview of the landscape of high-grade glioma and where we stand on treatments and where it's going, and the Q&A session will be at the end. Please bear in mind that panelists can't give specific advice to individual patient cases, but will do their best to answer all questions generally. And in a follow-up, we can address individual questions. It's truly an honor to briefly introduce today's incredibly distinguished panel. Dr. Sabine Mueller is PNOC Scientific Co-Founder, Professor of Neurology, Neurosurgery and Pediatrics at UCSF. She's an affiliated professor at the University of Zurich, Switzerland. Dr. Mueller serves as the principal investigator for a pioneering platform trial targeting diffuse midline glioma, among other research endeavors. Dr. Mueller has been inducted as the executive co-chair of the Children's Brain Tumor Network, a global entity dedicated to the aggregation of specimens, imaging, and data for pediatric and young adult brain tumor research. Dr. Michael Prados is a PNOC Scientific co-founder, Charles B. Wilson Endowed Chair in Neurological Surgery, Professor Emeritus, Department of Neurological Surgery and Department of Pediatrics at UCSF. Dr. Prados has over 35 years of experience at UCSF in treating both adults and children who have brain tumors. Dr. Andrea Franson is a pediatric oncologist in the Department of Pediatrics at the University of Michigan. She received a bachelor's degree with the Rensselaer Polytechnic Institute in Detroit, New York, and a medical degree from the University of Michigan Medical School. She completed a residency in pediatrics and fellowship in the Pediatric Hematology Oncology at the Children's Hospital of Philadelphia. During fellowship, she earned a master's degree in pharmacology from Thomas Jefferson University in Philadelphia. Dr. Franson has focused her research efforts in translational science, working to transition the most promising compounds into the clinic for children with cancer. Dr. Tom Bell-Davidson is a pediatric neuro-oncologist who specializes in caring for children with brain and spine tumors at the Children's Hospital of Los Angeles. Her research is aimed at developing novel clinical therapies with a focus on pediatric high-grade glioma and immunotherapeutics. Dr. Davidson has broad clinical and translational experience in running pediatric and young adult clinical trials. Dr. Jasper van Loot is a pediatric oncologist with a focus on high-grade pediatric brain tumors. He obtained his medical degree at the University of Utrecht and his PhD from the University of Amsterdam. Since 2018, he is appointed at the Princess Maxima Center as the treating physician and lead of the Neuro-Oncology subdivision at the Trial and Data Center within the Maxima. At this position, he's responsible for implementation and development for early phase trials. His research focuses on clinical and translational cellular immune therapy for children with a CNS malignancy. He's a member of the Independent Ethics Committee at the University Medical Center at Utrecht. And last but certainly by no means least, Dr. Elias Sayer is an assistant professor of neurosurgery and pediatrics and a principal investigator at the RNA Engineering Lab at the University of Florida. He functions as the newly appointed director of the Pediatric Cancer Immunotherapy Initiative of the University of Florida Health Center and vice chair of the UFHCC Scientific Review Monitoring Committee. As a board-certified pediatrician oncologist, Dr. Sayer has extensive translational experience as a PI and coordinator on several human trials. His translational efforts are also focused on new pipeline technologies, including a novel lipid nanoparticle formulation that he pioneered for the immunologic treatment of cancer, currently being tested in canine patients with terminal brain cancer before translation into dedicated human studies. So as you can see, we have a really highly qualified, esteemed panel joining us today, and it's time to turn over to the panelists for today's webinar.
Hi, everyone. It's really an honor and a privilege to be here with everyone, and especially to sit here on this panel with this international group of experts in the field of pediatric high-grade glioma. We all have a passion to find better treatments and better cures for this disease. So we're going to start off today by some general background about pediatric high-grade glioma. These tumors are the second most common malignant brain tumors of childhood. They make up about 10% of all CNS tumor diagnoses in children, but unfortunately account for 40% of brain tumor-related deaths. The current standard of care at diagnosis includes maximal surgery, followed by radiation therapy, plus or minus chemotherapy. There remains no consensus on the best upfront maintenance chemotherapy. Many sites will use temozolomide monotherapy based on our adult experience in the STU protocol, as well as the Children's Oncology ACNS 0126 trial, while other sites use combined temozolomide with lomustine or CCNU, also based on a Children's Oncology group study, ACNS 0423. But clearly there's a huge need for clinical trials so that we can optimize treatment right at the beginning in order to get our best chance of cure. There's no standard of care at all when it comes to recurrent or progressive diseases. People will try re-resection, re-irradiation, different chemotherapy combinations, but there is a real push and recommendation by the field to try to give patients access to clinical trials, to try novel therapies, and to learn more about these tumors. Next slide, please. I did want to focus a little bit about how over the last 20 years, there's been really an exponential growth in how we molecularly understand these tumors. And what we've learned is that these are a heterogeneous group of tumors. And there are many different subgroups that have very different biological triggers and might need different types of therapies as well. Some of the main subgroups are the infant-type hemispheric glioma, which occur in our infants and youngest toddlers. There are diffuse midline gliomas that have H3K27 alterations that can occur anywhere in the midline in the brain or spine or in the pons as a DIPG. There are diffuse pediatric type high-grade glioma that are histone and IDH wild type. There is a diffuse hemispheric glioma, H3G34 mutant tumors that often will occur in adolescence. And then IDH mutant astrocytoma, which we see most often in young adults, but also can see in adolescents. I think it's important as we talk about these clinical trials and propose them that we design them in a way where we are really able to look at our results and see how it affects these different subgroups, because as molecularly different as they are, they may also need different treatments in the future. Next slide, please.
Great. So I have the pleasure of being able to talk about one of the recently completed studies that we will be fully presenting at an upcoming meeting, as well as hopefully publishing within the next 6 months to a year. So PNOC 008 is a pilot trial testing clinical benefit of molecular profiling in order to determine individualized treatment plans in children and young adults with newly diagnosed high-grade glioma. And so we'll go through some of the high-level results here. So the eligibility was for patients less than or equal to 21 years at enrollment, patients with newly diagnosed non-DIPG localized high-grade glioma, and then patients had to have adequate tumor tissue because there was lots of molecular studies done on the tumor tissue to understand what targeted therapies may work best. Patients received standard of care radiation therapy first. While molecular analysis was being done, a tumor board met to review once molecular data was back, and the tumor board recommended up to 4 FDA-approved drugs for treatment for that patient. Enrollment occurred between 2018 and 2023, total of 55 patients enrolled with 50 eligible. Due to some diagnosis or tumor tissue difficulties. This occurred at 12 participating PNOC sites with 29 patients enrolled into stratum A, the hemispheric tumor, and 26 into the midline non-DIPG. The median age or average age was 11 years with the range of 2 to 20 and a relatively even distribution of genders of patients enrolled. And then the most common molecular diagnoses, which Dr. Davison just went over, so this is not really a surprise, were these different histone mutant subtypes, as well as a wild type diffuse pediatric type high-grade glioma. And then the schema on the right is just going through kind of the timeline of what occurred and tumor tissue being analyzed, followed by radiation therapy and then targeted therapy. So these are the most recent analysis survival curves that we have for stratum A on the left and stratum B on the right, with a median survival of 26.5 months for stratum A and 21.7 months from the time of enrollment. The most common side effects or adverse events that we saw at the higher grades, grade 3 or grade 4, that were considered treatment related and not related to other non-trial interventions, were not really unexpected, actually, and the first 3 are all blood count related changes. So decreased neutrophils, platelets, and white blood cells were the most common, as well as anemia. And then you could see much lower were some liver enzyme increases, which is alanine aminotransferase and aspartate aminotransferase, some oral mucositis, so inflammation within the mouth, as well as fever and neutropenia. But overall, generally pretty well tolerated, even when these were given in combination. So the broad conclusions at this point, but like I mentioned, a lot of analysis is ongoing. There was significant patient, family, and clinician interest in this approach, and in particular in this trial, it had relatively rapid accrual. The personalized treatment recommendations were feasible in this method and in a timely way. And so far, from what we have been able to see and analyze, survival data in both strata are encouraging. And in terms of next steps, we have ongoing analyses with central radiology review of imaging, circulating tumor DNA measurements, as well as quality of life measurements. And we will be presenting this via a poster presentation in Philadelphia in June and July, and then manuscript submission planned for this summer. So move on to PNOC 34.
So today I want to discuss a concept that we are developing and are very excited about for newly diagnosed children, adolescents, and young adults with high-grade glioma using pegylated arginine deaminase plus radiation therapy and temozolomide. Next slide. So in order to give some background, we have to go back to some basic biology and talk about arginine. Arginine is an amino acid, a building block for our proteins, and is very important in all sorts of cellular functions, such as cell signaling, proliferation, division. All human cells need arginine, but cancer cells need it even more in order to continue dividing and growing. Healthy humans obtain arginine mostly from eating and dietary intake, as well as some protein degradation. And when arginine levels are low in the blood, most normal human cells, though, can make some more arginine from citrulline via two important enzymes. The one that seems to be most important is arginosuccinate synthase 1, or ASS1. What is interesting is that some cancer cells lack ASS1, and because of that, they are not able to make more arginine. So if arginine is taken away from the blood supply, those cells can then eventually die, leading to tumor cell death. Some tumors that don't have ASS1 are mesothelioma, non-small cell lung cancer, GBMs, and from our early data, it seems that some pediatric high-grade glioma also don't have ASS1 expression. Next slide, please. Now, arginine deaminase is an enzyme that breaks down arginine in the blood. ADI-PEG20 is a novel agent from Polaris Pharmaceuticals that is a pegylated form of this enzyme. And what that means is that it's able to stay in the blood system for a prolonged period of time to break down any arginine that might be available for tumor cells. This drug has shown in the laboratory in mice and in cell culture to be very effective against ASS1 negative cancers. This drug has also been explored in clinical trials for a number of different tumors and have been shown to be quite safe. And there are continued clinical trials using this drug alone and in combination with different chemotherapies. Next slide. This is a very busy slide, but I think it's important. So there is research out of a lab in England by Dr. Saeed, who's looking at the combination of ADI-PEG20 and temozolomide in adult GBMs. She had experiments where she had mice that had human GBM tumors that were either ASS1 negative, so they couldn't make their own arginine, or ASS1 positive. And what she's found is that the mice that had ASS1 negative tumors, when they were given ADI-PEG20 alone, had a decrease in tumor size and an improvement in survival, and when they were given in combo with temozolomide, that effect was even seen higher. But for the ASS1 positive tumors, because those cells can make their own arginine, ADI-PEG20 on its own did not seem to be effective. But when given with temozolomide, it had increased tumor kill and increased survival, showing that no matter what the ASS1 status is, the combination of temozolomide and ADI-PEG20 is promising to cause increased tumor kill and hopefully more survival in high-grade gliomas. Next slide. The same group looked at the combination of this drug with radiation, and in her mouse model, she found that ADI-PEG20 plus radiation completely obliterated the GBMs in the mice and had very prolonged survival. Next slide. So based on this data and some early pre-published pediatric data, we are proposing a pilot study for newly diagnosed pediatric high-grade glioma. There will be a safety portion and then an efficacy expansion. We plan to add ADI-PEG20 to a backbone of temozolomide and radiation therapy, followed by maintenance of temozolomide with ADI-PEG20. We will have 3 cohorts: the histone wild-type pediatric high-grade glioma, the H3G34, and the H3K27 mutated gliomas. And again, having each their own cohort so we can see how this approach works in all these different groups. Next slide. The primary objectives will be to look at the safety and the recommended combination doses, but also to look at the efficacy and anti-tumor activity by looking at 12-month progression-free survival. Next slide. Eligibility will be all patients with histologically confirmed, so post-operatively newly diagnosed high-grade glioma with either wild-type H3K27 or H3G34 mutated, and patients will be eligible if they're age 3 to 39 years of age. We will require tumor tissue, and patients should not have had any prior therapy. And the only exclusion, the most significant exclusion criteria are allergies to pegylated compounds, or unfortunately, we will not include diffuse intrinsic pontine gliomas in this study, although there are other PNOC studies for that population. Okay, moving on to the next study. This is a study that's actually open and open to enrollment. This is, instead of for newly diagnosed high-grade glioma, this is for recurrent or progressive high-grade glioma. PNOC 19, neoadjuvant checkpoint inhibition. Next slide, please. So a little background on the immune checkpoint inhibition. The immune system has checkpoints or safety blocks to inhibit ongoing immune reactions. That way, if there is a normal immune reaction in the body, it doesn't continue indefinitely and cannot hurt normal tissue. Within the tumor cells and the immune microenvironment of the tumor, the tumors have optimized this human's checkpoint functions to increase those checkpoints and prevent the tumor from being recognized and killed from the immune system. Drugs that block these checkpoints or safety points can turn back on the immune cells and potentially recognize cancer cells and cause cancer cell death. Next slide. One of those checkpoints is programmed cell death, or PD-1. PD-1 is expressed on T cells, which is an immune cell, and binds to PD-L1, which is often expressed on tumor cells. When these are bound, the T cells no longer can attack the tumor cells. So there are a number of different PD-1 inhibitors or monoclonal antibodies that stop the PD-1 from binding to PD-L1. When this happens, it boosts the immune system back to being functioning and potentially can attack cancer cells. We have seen clinical efficacy in this approach in a number of tumor types, such as melanoma, non-small cell lung cancer, and renal cell carcinoma. Unfortunately, the early studies in high-grade glioma were not very successful. But because the science was there, people started exploring, what if we give it in different schedules or in different ways? Maybe we can exploit the use of these monoclonal antibodies. Next slide. So a few years back, there was a small pilot study through the Ivy Foundation for adult high-grade gliomas, where they wanted to see if you give the medicine prior to surgery, does it enhance the efficacy of these PD-1 inhibitors? So in this study, in recurrent adult GBM, patients were randomized to two arms. One group got a single dose of a PD-1 inhibitor, pembrolizumab, prior to surgery, while the other group got no medication prior to surgery. Both arms had surgery. We collected the tumor to see what was happening in the microenvironment, and then everyone got PD-1 inhibitors following. Next slide. What the team found when they looked at the tumor was, in summary, there was an increase of immune activation and T-cell activation in the patients that received the single dose of PD-1 inhibitor prior to surgery. Although it was a small study and not really powered to look at survival, what they also found was that the group of patients who had that single dose of PD-1 inhibitor prior to surgery had almost a doubling of overall survival. Next slide. Based on this exciting data, we developed a study for recurrent or progressive pediatric high-grade glioma. This study is a single cohort study where all patients at recurrence and progressive who enroll in the study who have operable disease will receive a single dose of PD-1 inhibitor, in this case nivolumab, prior to surgery. We will collect the tumor, again, to interrogate changes compared to historical tumors that have not received checkpoint inhibitors. And then everyone will receive the PD-1 inhibitors following. Our primary endpoints is really to look at the tumor environment and see how it's changed with these agents, and also to characterize the safety of this method. We will also look in secondary and exploratory endpoints how this affects survival. Next slide. Eligibility will be patients who have histologically proven high-grade gliomas that have already had first-line therapy, that have had progression on MRI. It's open to patients 6 months to 25 years of age, and they must be eligible for surgical resection. And it must be at least 12 weeks from prior exposure to PD-1 or PD-L1 inhibitors. And the main exclusion criteria, again, are patients who don't have clinical reason to have surgery. Okay.
So I think I continue from here. So thank you very much for allowing me to speak. I'm very excited about it. I will go over the highlights, so we have enough time for some discussion at the end. So firstly, a study of Dean Lee and Mohammed about NK cells in young children with a supratentorial malignant brain tumor, and amongst them obviously are high-grade tumors. And NK cells are a very interesting group of immune cells. There are lymphocytes as well, but they don't need an antigen to be active, so they can kill without having this really recognition with other T cells really need, and therefore they can be an interesting way of immunotherapy because they're less labor intensive than CAR Ts, for example. You can use them off the shelf ideally, and they play a very important role. We learn more and more in tumor immunology. So therefore, this is a trial we want to see if NK cell actually can be a useful immunotherapy. So next slide. So what is done in this trial? The NK cells of a healthy donor are growth on a cell layer to expand them, and they are giving some factors, TGF-beta, for example, you can forget the name, where they mimic more or less the microenvironment, and therefore they are trained to survive in this certain kind of microenvironment of the tumor. So hopefully they can persist and stay alive for a longer period once they are in the tumor. This is what happens in the trial. There is a tumor, there's a resection. In the resection cavity, there they place an Ommaya drain, and here on the picture you see an Ommaya drain which is placed infratentorial, but it will be supratentorial, and there they will administer those NK cells on several time points. So it will be multiple injections of those NK cells in this tumor cavity. Next slide. Patients, as said, need to have a supratentorial tumor, high grade, grade 3 or 4. So that will be high grade gliomas, but also, for example, ependymomas. And they also need to be a candidate for the Ommaya reservoir, because it needs to be after in the cavity of the tumor after surgery. There needs to be enough place to really administer those NK cells. Next slide. So the objective is about safety and also seeing what's the best dose to move forward to the next phase of this trial. Next slide. Yes. So in summary, so the PNOC 028, I think is very interesting. You have NK cells administered directly at the tumor sites, and if this works out, it's also much cheaper and less labor intensive, and you can maybe treat more patients at the same time than you do for CAR T trials. So as this webinar is really focused on immunotherapy, I want to touch very shortly upon two other ways of immunotherapy, being the oncolytic virus. You may have heard of it. It's a virus that is modified, that is like infected only the tumor cells. And now what is done most of the time is injection directly into the tumor. There it starts infecting, as you can see here, the tumor cells. And because of this replication, these tumor cells will die. Then you get a release of all kinds of proteins, etc. And because of that, you get an activation of the immune system. And with activation of the immune system, you hope to have a second hit that the immune system starts cleaning up the rest of the tumor as well. So both locally, but also systemically. Next slide. So this is how it's typically done at the OR. The patient is planned to get the infusion of the virus. And then you get here 2 cannulas injecting the virus in 1 hour at the OR. And after that, you get some contrast fluid so that we actually can see if the virus, if you can see in the last picture, you see a white spot. So we know there we placed the virus, so that's in the spot we wanted. So it's a good control mechanism that we have injected the virus. Next slide. Yeah, so there has been one trial performed in the IPGs. I think this picture of the tumor is not really fair because it's also after radiotherapy. But what you do see is that there was a longer survival median over 17 months, and up till now there are still 2 patients alive. One of them is a K27M mutated, another one is a wild type, so that's a somewhat different tumor. But you do see what you see more often in immunotherapy trials, you cannot get off a tail at the end in your survival curve, meaning you have some longer survivors there, but also a lot of non-responders. Next slide. And again, most of you heard about CAR T-cell therapy. This is a way of getting your T cells out of the patient and modifying it against a certain target in your tumor, and then give this product back to the patient, either IV or directly into tumor, or what's now mostly done intraventricular. And the most recent paper in adults of glioblastomas just came out, again with some strong signal that it may be beneficial or a good way forward. All transient, but you did see a clear response. It was against EGFR, which we don't see as much in pediatric high-grade gliomas. For example, here at the second, do you see my mouse actually? You see here they started to administer the CAR Ts, and already after 1 day you saw a good response. Again, in the end, it came back as well, but you do see a signal, so it's a good start. So next. Yeah, so I think we, as the PNOC community, very much advocate for strong correlative studies. So we really want to learn as much from each individual as possible, because if the treatment is not successful, at least we learn from it and know hopefully why patients do respond and do not respond. And it really asks a lot of commitment from patients and families, because you have to collect blood, all kind of other things, but then we can really start to learn more and more. Next slide. Yeah, so and now more and more we dive into the microbiome as well. So there's an example. So the next slide. Oh, here's the next slide. Yeah. So the gut microbiota, that is, I think, more and more we know it plays a huge role in all kinds of different metabolisms. It can predict for toxicity, and even for some trials for efficacy, has a role in inflammation. So we don't know much about pediatric brain tumors and microbiome. We start collecting those data and to see what the role can be and if we can modify those microbiome. The next slide. Yeah, so this is one example. I don't go over it in detail, but it's even already at the stage that for melanoma patient, they started to do fecal transplantation after a patient was resistant to a certain type of immunotherapy, checkpoint inhibitor. Tom was just telling about. They did feces transplantation, and 50% after that start to respond again, because they did fecal transplant of patients who did respond. So these, I think, are very interesting things which can come out of correlative studies. So next. slide. Yes.
Well, good afternoon, good morning, everybody. Thank you for having me present today. I'm going to present our work with RNA vaccines. And as we get started, I just want to say I'm profoundly sorry to everyone here who's been affected by this horrible disease. PNOC is really filling a fundamental gap. And while the pace of progress is slow, there has been significant progress. So our work at the University of Florida has really involved developing new therapies. We're hearing about a lot of new therapies. Our therapies are more focused on mRNA vaccines as immunotherapies. And the field of mRNA has really evolved over the last few years, post-COVID. There are different technologies that actually have made their way into the clinic for cancer using mRNA. The original vaccines were these protamine complexes that were administered to patients. And this happened really at the early part of the millennium, the first 10 years or so. And they've been replaced with what we're familiar with now, these RNA LNPs, these lipid nanoparticles. This design has been used in the COVID-19 vaccine for spectacular success and is being used for melanoma patients. So there is a randomized placebo-controlled phase 3 trial from Moderna using these RNA LNPs for patients with refractory melanoma. Now, while that's really promising, melanoma is what you'd refer to as an immunogenic tumor. Okay, those tumors elicit a really strong immune response. And that just isn't true for the bulk of cancers, certainly for pediatric cancer. And that really leads me to these next two technologies. BioNTech has recognized this, and they've developed a different formulation. It's called these RNA anionic lipoplexes. And unlike the LNP approach, which is given as an intramuscular injection, this is given intravenously. And it's given intravenously really to elicit a very rapid and strong immune response. And this needs to happen rapidly, within hours, because cancer is evolving. Now, our group has taken this a step further. One of the biggest challenges of packaging RNA into a lipid particle payload is you just can't package that much. RNA is negatively charged, so if you put too many negatively charged particles in a really tight space, you're not going to have that many in there. And we know that the more you add, the more immunogenic that compound is. And so we've developed these multilamellar lipid nanoparticle aggregates that I will talk to you about some more. Next slide, please. So we have found that these multilamellar particles are not just single particles, but they're clusters of particles. And the real premise for this work was really trying to take someone's tumor and make it look like an active viral infection. And interestingly, one viral particle is not as immunogenic as many viral particles stuck together. So if you had 5 individual viral particles, that would not be as strongly immunogenic as those 5 particles stuck together as a cluster. And so we've developed these RNA lipid particle aggregates, these clusters that look a lot like onions stuck together, where we have a lot of the patient's own tumor RNA. And the beauty of RNA is from just 100 biopsied cells, we can extract all the messenger RNA and amplify it, having a renewable resource of that patient's own tumor information that we can repeatedly vaccinate against. Next slide. And so we've tested this approach in many different mouse models. So in the top row, we've seen improved survivor outcomes in mice with osteosarcoma, mice with adult glioblastoma, and we've also seen responses in mice with the more pediatric diffuse midline glioma expressing H3K27M. And we've shown we can vaccinate these mice with either the entire RNA of the tumor or really make it against something very specific like H3K27F. We've advanced this into dogs with gliomas, dog glioma. These are pet dogs that have natural forms of cancer. They're enrolled on their owner's consent. These are really difficult diseases in dogs, dog glioma. Canines die usually within 1 to 2 months with palliative care. And we treated 10 of these dogs with whole mRNA from their tumor, just giving them these vaccines, 3 vaccines, and saw a median survivorship of 139 days. We have now advanced this into human trials. So we've treated 4 patients, all who have lived longer than expected. But I want to draw your attention to one patient who had what we thought was tumor progression. And we biopsied that tumor, and there were no tumor cells. And what you're seeing in red here is really immune cells. So that was very encouraging. Now, we've been moving this a little slowly into pediatrics. One reason is we want to make sure we're doing this safely. These patients did have immune effects, fevers. They had some of what we refer to as cytokine storm, meaning the immune response is so active that you have to really monitor these patients carefully, sometimes in the ICU, to make sure that the immune system is not causing really significant deleterious effects. Next slide, please. As we move this forward into pediatric patients, we've really had the good fortune of partnering with the Pediatric Neuro-Oncology Consortium. PNOC has invested in this before the COVID-19 vaccine, so I really want to give credit to the consortium and its scientific membership for investing in this long before RNA became very contemporary. And we're moving this into a multi-institutional trial through the consortium, and really developing a lot of the manufacturing queue, cold chain that BioNTech and Moderna had to work out for their COVID-19 vaccines. And we've certainly made a lot of progress in that regard. This is for patients with primary high-grade glioma. We would be getting a biopsy tissue from these sites sent to us to manufacture the vaccine. By the time a patient finishes primary therapy with radiation, we'll be able to send those vaccines back and really just treat the patient with immunotherapy. Next slide, please. So I think while the pace of progress is slow, there is a lot of hope for immunotherapy. I think I can really just summarize it very simply here with, you need a dendritic cell. A dendritic cell is like the general of the army. And so you have to teach the general of the army how to fight the invaders, either a virus or a cancer. And now that general of the army has to go teach the army members, and those are the T cells. And now we are seeing across PNOC several different approaches that can harness the generals, dendritic cells, there are other generals, macrophages, other antigen-presenting cells, but really kickstart a response. And we're also seeing approaches where we can basically prime the T cells, get the army members more educated, either through taking them out of the body, engineering them and giving them back, or through other means, drug therapies, etc. One of these drug therapies that you've heard about from Dr. Davidson is checkpoint inhibitors, really to maintain these cells in the on position. And you're seeing this play across PNOC. We have several PNOC trials for vaccines, nanoparticles, adoptive cell therapy, immune checkpoint inhibitors. And if we can just think to the future, as we get these trials to the point of feasibility, safety endpoints, can we combine them? Can we combine them and create a combinatorial immunotherapy approach that mimics what we do in leukemia with an induction regimen, using a couple of these things together to really orchestrate an immune response in a way that's self-sustaining, and then consolidate it. Don't stop there. Keep going, consolidate it with one or more of these therapies before eventually maintaining it with checkpoint inhibitors or other therapies. So this is how combination chemotherapy started. Single therapies were not very effective, but those trials were critical to run to show effect, meaning you could get some benefit, even if it wasn't long-lived. And as that data was acquired through multiple different single agents, the combinations came to pass. Next slide. So PNOC is incredibly primed to have a paradigm much like this as we launch these trials, followed by secondary trials looking at combinations. But PNOC has also established infrastructure through the Children's Brain Tumor Network. We have developed immunomonitoring approaches that are becoming centralized through Jasper's group, through partnership with Children's Hospital of Philadelphia and other groups. There's new AI approaches to really help us predict which patients are responding, which patients are not responding. How do you know you're getting an immune response in some patients? We want to be able to tell a patient as soon as possible, hey, this is working or it's not working. If it's not working, let's get them on another trial. If it is working, let's keep going. And so in this way, samples we collect from patients, whether it's tumor, whether it's blood, whether it's CSF, are moving the field forward. We're making progress from every patient we treat on trial. We're making progress from every trial we run. Next slide, please. And ultimately, it is these small steps, and I do apologize that they are small, but these steps are leading to transformational growth. And I think this is emblematic of our own PNOC 20 trial. We have not treated a child yet with these RNA vaccines, but the infrastructure that we've developed to get stable vaccines from one institution to another, this cold chain, that infrastructure now can be used for any trial, not just PNOC 20. It could be used for an osteosarcoma trial, another brain tumor trial. And so those small steps can really lead to the transformational growth, the transformational change I think we're seeing. And that future, I think, is quite hopeful. So thank you for your time, and I'll stop there.
Great. Thank you. Thank you, everyone. Wonderful speakers. So there's a question. I will start with the question in the chat. So Tom, this is for you. For the PNOC 34 trial, the IDI-PAC, how do you think about sort of dietary arginine and how that potentially will affect outcome?
So that's a great question. There's been quite a bit of human data already on clinical trials using this drug and other tumor types. And what we've learned is that ADI-PAG20 is very efficient at breaking down arginine to unmeasurable levels. And that's despite a diet full of arginine or no arginine. So we feel pretty confident that we don't need to do any sort of restrictive diets on this therapy, but it's, something we should continue to look at as we are designing all these studies.
Great. Thank you, Tom. Another question is, how do we decide within PNOC what ages are allowed for PNOC trials? So that's a very good question. I don't know, Mike, if you want to answer this, if you want me to answer that.
I think I'll just take a quick stab at it. I think the vision of PNOC is to develop therapeutics for children and young adults with high unmet need. And there's a group of adults we call the AYA that are very poorly represented in consortia that do clinical trials for them. So I think they're left out in some ways of a lot of trials. So they're just as important. Somebody who's 28 or 30 is just as important to us as somebody who's 2 months of age. So at least the vision, I think, of PNOC is to include all of those patients and to learn from them. They're very uniquely different in terms of the biology of their tumors. And we have to keep looking at their tumor to understand their biology and to design therapies. I think it's totally appropriate for this consortium to be doing young adults.
Yeah, and I would say, so how we determine this, I think the intent is to be as inclusive as we can, and then obviously match based on age sort of responses and toxicity. I would say that sometimes our industry partner or also regulatory restrictions limit us in this intent. Sometimes, even if you would like to go down to age 0, 1, or 2, that's sometimes not allowed. Sometimes we need to start our trials in an older population, often 12, 13 years of age, before we can go to the younger population. Sometimes industry has efforts already in the adult setting, and so they are restricting us then to basically hold enrollment up to 21 years of age. So there are limitations that are not necessarily scientifically driven, and we just have to live in this reality. Elias, there's a question for you from the audience. What do you think the timeline is for PNOC 20 that children are enrolled?
Well, we're anticipating this year, hopefully, if all goes well. Third quarter, fourth quarter of this year is our anticipated start date.
Yep, great. Yeah, that's definitely our intent. And that was another mandate from the FDA to start first in the adult population, and then before we move it into pediatrics. And in pediatrics, the additional challenge we faced is it's obviously much simpler for such an exciting therapy to do a single institution trial. We're a multi-institution. There's much more quality or control and a lot of things Elias really and his team had to work out. And so that's why often we see, unfortunately, still delays bringing these novel therapies to our children. There's another question about under 3 years of age. I think we answered this when we discussed sort of age restriction. We like to include them. I think there's specific consideration, especially in the very young patients, but we often want to include, and then it's sometimes just restriction based on regulatory regulations. There's another question. And maybe Jasper, that's for you. Does anyone has experience of oncovirus delivery through Ommaya reservoir intracerebrally? And how do we think about it?
Yeah, so that's a very good question. And on one hand, it sounds very attractive because you can have repetitive injections, but there's also a concern of encephalitis in those patient, that you get inflammation of the virus itself outside the tumor itself. That's what has been seen also in animal models. So that needs to be a bit more testing, and it also depends a bit on the virus, because you have all kind of different viruses. You have the adenovirus, herpes virus, etc., and they all have a bit their own dynamics. So the field is a bit reluctant to give it through the Ommaya, because you can have kind of meningitis or encephalitis, although it's supposed to be selectively infect the tumor cells. They are afraid that you still get some inflammation. So it is a very interesting, I think, approach, but we need to get a bit better idea how safe it actually is.
Yeah, yes, but thank you for that answer. And I would just add, we have completed a clinical trial using sort of attenuated measles virus for embryonal tumors. And in that setting, we gave the virus either directly in the tumor bed, but we also gave it not through an Ommaya, through a lumbar puncture, so also disseminated. But we've done a lot of preclinical studies making sure that we don't in animals see this type of sort of inflammation of the brain structure surrounding your brain, what Jasper mentioned, like meningitis, encephalitis. But in our trial, giving it directly into the cerebral spinal fluid, we have not seen any concerns about encephalitis. So it might be really also dose and virus dependent, but you need to provide the preclinical data to make sure that you're obviously not causing these effects, what Jasper mentioned. I think that's a really challenging question, but maybe one of you can attempt to answer it. So one of the attendees asked the question, if a low-grade oligodendroglioma, can that change into a GBM? And if we think about that transition, which I think we often see more in the adult setting than in the pediatric setting, that low-grade glioma can potentially progress to high-grade lesions, how can we potentially prevent this? What are the thoughts? I don't know if anyone can easily answer the question, but Elias, go ahead.
I think
Yeah.
there are IDH mutated low-grade gliomas that have been in the news a lot lately, because there's been a few large adult trials that have showed great efficacy in IDH inhibitors. Those trials have been inclusive of adolescents, but there haven't been that many adolescent patients on those trials. So there's still a lot to learn about how those inhibitors affect younger patients. We know that in adults, IDH mutated tumors do eventually transform into high-grade gliomas. We actually have much less data in the adolescent age group. It is possible that it is part of the same disease and just eventually acts similar to adult tumors. But I think the point is that we need to do research in the teenage population with IDH mutated tumors, and something PNOC should think about.
And I think there is an opportunity for vaccines here. Now, no one's going to let us test these things in kind of lower grade settings. They have to be treated in kind of the most refractory settings first. But if we are to demonstrate feasibility safety, for example, in high grade settings, could you make an mRNA vaccine targeting an IDH mutation, a BRAF mutation, a BRAF fusion, other fusions, and actually use that as a secondary prevention approach, meaning, tumor's out, and now you can kind of just prevent, you'll get the straggler cells through the immune response. So I do think there could be an opportunity for secondary prevention from vaccines, maybe even primary prevention one day, but is very challenging right now.
Great, yeah, thank you. One of the questions from the audience was about what are sort of the key areas where, as a community, we really would need more funds to really complete it and be faster. And so I think sort of the key needs are, as always, and I think Elias made really a wonderful point, it is also in how can we use technology that is being developed in other areas to better also understand responses and failures in our pediatric neuro-oncology community, especially in high-grade gliomas. So looking at AI, machine learning investigations, how that could help us to predict responses, failures earlier, all the correlative studies, developing these large model systems where we take into account the characteristics, the phenotypic and genotypic characteristics of a patient and the tumor, looking in much more detail than we currently can by just looking with our eyes and neuroradiology at imaging, and then all the wonderful correlative studies that also Jesper mentioned. And putting that all together is a tremendous amount of work, really, just in terms of a platform development. And we are collaborating with CB10, but it still takes a lot of manpower to get every scan correctly labeled so that people really can make sense of it. So I think there's always a need to support these larger data sets efforts, which I think will be the key for us to really then understand much more in detail sort of responses, failures for our patients. There was another question. I know we are slightly over time already, but we will continue to hopefully answer most of them. So I think, Jesper, maybe you could address that. So is it possible to combine reirradiation and oncovirus therapies, and what has been sort of a current experience with that?
Yeah, so the trial that has been done in the IPG was in combination with radiation, so that is safe at least. For re-radiation has not been done in that setting, but I anticipate that it's safe as well, as long as you anticipate upfront. But I think that holds true for all immunotherapy trials, that you have enough space to have some pseudo progression. So you have some inflammation of the tumor that can happen because of immunotherapy in general, but also oncolytic fibers, which are directly infused into the brain. But also re-radiation itself can obviously give some swelling and pseudoprogression of the tumor. So you really have to estimate before you actually administer the virus that you think you have enough space for this. So, but I think it's safe because it has been done in combination with radiation therapy.
Great. And so one last question, which I think is always a very important question. What is the best way how a parent can find a clinical trial? And so I think, it can be very, very overwhelming. All the trials are registered on clinicaltrials.gov, but it's sometimes really challenging understand, is a trial really actively accruing? Is your child really eligible? So I think the best way is really to have a close communication with your provider around that. So they should be in the best position. I don't think I would be shy if my child would be diagnosed with a brain tumors to also ask for a second opinion. I think it's crucial to really develop your team. Sometimes it's really helpful also as a family. You are so occupied and busy taking care of your child to have maybe either a relative or a friend helping you in contacting different sites and centers to help you just navigate that landscape. But it should be really in close collaboration with your primary team, and they should really provide you that. For some of the entities like diffuse midline glioma, some families came together and they established a national diffuse midline glioma tumor board where every child can be presented, and it's a free service. But then really the key experts running trial for this entity are on this call reviewing everything and then can make a decision if a child is eligible or not. That's not accessible and globally in place for other entities. But so that is one forum where if you have more like a diffuse midline glioma that can be developed. But obviously, if there's current initiatives that would want to establish something like this for other tumor types, we would be obviously more than happy to collaborate. But my strong advice would be develop a team, assign tasks to your friends and families who do want to help, and don't be shy to ask for a second opinion if you feel like you don't get the answers from your treating team. And so I think with that, we will end. I want to thank all the wonderful speakers today. Obviously, this will be made available online, right, Rachel? And then if there's any other questions or further thoughts, never hesitate to reach out to us. Our information is on the internet and email. And thank you very much for joining today. I don't know, Bruce, if you had any last comments, but otherwise, I think we will close. You're on mute, Bruce. We can't hear you.
Thanks for being the dreaded mute. Look, as a patient parent who's been in this space for over 20 years, I do understand that it's frustrating that things aren't moving faster, you know, if you're a parent or a family who's in this space. But I will say that I've never seen more promise and potential. The power of the collaboration that you guys are bringing to this space is really, really, truly amazing. And I think we're at the foundation dedicated more than ever to aggregating funding to continue to drive this progress. I think the only thing that's holding back, you know, acceleration in this space is resources. So I think it's great for everybody to be aware that the more funding that we can bring to this, the faster that we can actually move forward. And I applaud all of your selfless efforts in actually helping these families that are, you know, have been involuntarily inducted into this, you know, pediatric neuro-oncology space. There is hope. Let's keep going. And thank you so much for all of your efforts and for your contributions today.